So basically, what is histology? And it's derived from the Latin for the study of tissues. Okay, and so I don't know. Give me an idea. How many of you have taken AP Bio? Can you raise your hand, or in AP Bio, raise your hand here? Okay, actually raise your hand if you're familiar with what a cell looks like, at least like a diagram, right? An animal cell versus a plant cell. Is everyone familiar with that? Can you guys raise your hand? Does that work for you? Okay, I see Anushka, Samira, Amaya. All right, there you go. Good. That's good. That's a good fundamental to go here. Awesome. Thank you guys. So when we talk about histology, it comes down to, if you're familiar with the term biopsy, maybe you know the word, but not exactly sure what it is. But basically, in a procedural setting, or maybe you hear it just like with a dermatologist, for example, might take a biopsy of your skin to see if there's a cancer. Okay, what does this actually mean? What they're doing is they're taking a little bit of tissue, and tissue just refers to, for the sake of argument, a collection of cells in a given area. And so in different organs, you have a different tissue makeup in the sense that the cells are different, they operate differently, but the fundamental understanding of what a cell is still applies, right? And I'm sure you're familiar with this concept of cells making up everything, right? That's quite straightforward. And so when you take a biopsy, to understand both pathology, what is wrong with a patient, one way you can do that with certain illnesses, and we'll see this today, is to take a biopsy and look at the tissue under a microscope, where you can actually see the cells and see certain proteins, or at least representations of proteins based on dyes. And so one thing, when we start to look at these, you got to know that cells themselves, you aren't able to see that with visible light, right? And you know how vision works, right? Light has to bounce off of something for you to be able to see it. But cells don't have color per se by themselves. And so what we use are dyes to actually stain specific parts of the cell that we want to look at and know. And this gives us a bearing when we're actually looking at this at these tissue samples to understand what's going on. And so don't worry too much. I'm not going to go into the chemistry of it in too much detail. But you see on the right, you have basophilic and you have acidophilic. All right. And basophilic is in purple. This is key. The colors are key here. So these specific dyes, and they're standardized. They're certain specific names of the dyes that are used specifically to bind to DNA, RNA, and sulfated polysaccharides. Don't worry about that. But think about, where in the cell is there going to be DNA and RNA, right? Does anyone give me an idea? Where would the most DNA be in a human cell or an animal cell? See the chat. Exactly, right in the nucleus. And so when we start to look at these, you'll know exactly where the nucleus is because you'll be looking for that purple color. And then you have the reddish color, which is the proteins. And so this one's a little bit more abstract because proteins are everywhere, right? They make up the cell. And so you'll begin to see with certain cells, certain cell types, more proteins will be present. And we'll talk about that in more detail. I don't want to kind of talk about it too much right now. Okay, so you have some bearing for what we're looking at. Okay, so here is a tissue sample from the liver. Okay, so a human liver. And so we can zoom in and actually see what the cells look like inside. And this is the fundamental of what histology is. Whenever you're looking at it, you're going to be looking at something that it won't necessarily look like this, but when you zoom in, you'll see those individual cells, and they'll all look different depending on what organ you're in, what tissue you're in. But here you can see the example of the liver. And so I want everyone to see and sort of identify what they're looking at here. All right. Okay. We have two colors. Right, we have that pinkish color, and then we have that purple color of the nucleus. Okay, and so this should give you some bearing whenever you're looking at slides. You have your cell wall kind of surrounding. I don't know if you can see my cursor. I have a bigger one here. You have your cell wall kind of surrounding. You know what the cell wall looks like of an animal cell. It's kind of round when we look at the diagrams. And then you have your nucleus in the middle, right? And so every time, be aware what you're looking at in that regard. And then just so you know, when you take a biopsy, and when you're looking at something on a slide, and you've done this in your biology classes, right? You need a very flat, a flat level sample, right? What you're looking at has to be flat and cut. And so when they do this, they have to have a very thin slice of tissue. And that's why you have, we can't cut it. We try our best to cut it as specifically like a 1 cell thickness, but obviously that's not, impossible to achieve with the technology. And so that's why you'll see some overlap. That's why it's not exactly cell, 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 as you might imagine. But in a plant cell, you might see that because you have the cell walls that rigidly fit together. Okay, so let's go into it. Here's our first kind of sample. Someone give an idea. I don't know if you can see it on the side, what we're looking at, but this is the spinal cord. I was going to ask, but it says it already. So the spinal cord. And I think, one of the coolest things to look at here is to understand the difference between different parts of this sample. All right, so you can imagine kind of a cross-sectional cut where here in the middle, can anyone give me an idea of what exactly this here in the middle might be? Either unmute, raise your hand. Come on, I could talk to you direct. I could just talk at you, the entire meeting. But if you guys want to kind of try to answer questions, that's okay. Oh, nope, nothing in chat. So this is a spinal cord, and surrounding the spinal cord is, what do you think? If you're taking a cross section, maybe for those of you who saw the CT scans from a previous meeting, what would surround on both sides? We even talked about this at the last meeting with neurology. Okay, it's the vertebrae. It's the vertebrae surrounding the spinal cord, which protects it, as I'm sure you know. And so I want you to kind of look at and understand how many different types of cells exist just in this single cross section. This, you can actually look in the bottom left and you can see how large it is. 1,000 microns or micrometers, same thing. And so to give you some perspective, 10,000 microns is 1 centimeter. So it is, extremely small. And as I zoom in, you can see that scale changing. So you have some perspective for how big a cell is, right? And so you see again, this purple color is our nucleus. And we have, 80 microns is this, a whole distance here. And each cell is only a very small portion of that. Okay, so one thing to take note of is here you have your vertebrae. What do we need to know about this? You have spongy bone tissue here, okay? And so maybe you're familiar with the diagrams of the long bone, and on like the ends, you have kind of a spongier tissue, and then you have your bone marrow in the middle. The actual cells that are there are very different. And so they look very different on histology. And so if there's something wrong, you can make a differentiation between what is this? This is normal, right? From what a pathologic example could look like. And then out here, you have muscles and you have, think about your body, right? Think about, I can't remember, I think this is a cervical section. So right near your neck area of this tissue sample. And so you want to keep in mind, what is there? You have muscle, probably have fat. That won't be, a super part. You'll have a super big part of this. However, you could see adipocytes, which are like usually show up as large and white because they don't have a nucleus. You don't see that here, but you see these kind of these cells, which are muscle cells, all right, around, which makes sense, right? You want that to be around your vertebrae. And if you can imagine, if you feel your back, you have, around your vertebrae, you have muscles that are going to be protecting you. Okay, I'm going to jump to the next one. The eye is a little bit more complex, and I won't go into too much detail, but I wanted to show this because I know, those of you who got to do the eye dissection last year kind of get some perspective on what it would look like. And this is the cross section, of course. And you can see, you have your optic nerve back here. And so we'll talk about it in a little more detail. But here, I want you to know how certain regions here don't have cells present. And that one of the really specific, so here you have cells across the front portion here. which makes sense because it keeps it structured. However, here we have the opening, right? This is our opening to the lens. This would be our pupil. And then here we have no cells. Can you tell there's no purple nuclei that we would be looking for if we were looking for cells. This is your lens. And now, can you imagine, if we had cells in the lens where we want to be able to, see from and have light penetrate through perfectly well, right? And so that's why the lens is this way, okay? One thing also is very important is the iris. If you remember when we talked about the iris being what actually opens and closes your pupil, right? And so that's how your eye dilates or constricts, whether you're in a high amount of light or a little amount of light. And here you can see that this is a highly vascular region. There are a lot of cells here, and I think really the big thing is the cells here. And you'll see this coloration that's a little bit different. And so there's a different dye that's used specifically for I couldn't tell you exactly, but I know what we're going to look at next is that there's a darker kind of brownish dye that's used for the Golgi apparatus, which I hope, I think that's interesting that you can see now that different organelles are being focused on and accentuated when we're looking at these histologies. I can't guarantee you for this one because it's not explicitly in the statement and I don't want to give you wrong information, but just so you have an idea of what that kind of looks like. And then one thing I also want to show you is that this optic nerve in the back here, notice how all the cells kind of line up and are directional in a sense. You're going to see what I'm saying there, as opposed to kind of like our liver sample, where it was kind of hodgepodge in a sense. It was kind of all on top of each other. And so you want to see, think about, all right, what is the optic nerve? Okay, it's a bunch of neurons, which makes sense. And neurons all are adjacent and connect. And so the way it's represented here with the specific dye, when I say represented, I'm specifically referring to the dye they chose because it'll look different with different dyes. But you can see that kind of directional presence, right? And which makes sense if we're thinking about the optic nerve. Okie dokie. So here's the next one. Here's an example of the esophagus, all right? And so if we zoom in here, we can see, there's a ton of different cell types per se, right? Here, because you have cells that are more, the orientation, you see that kind of longitudinal or more like lateral as opposed to these that are more clumped together and much, much smaller in terms of how they're compressed and the way they make up the esophagus and surround that opening of the esophagus, which is this big white space, okay? And then you'll see again the muscles around here, and maybe you've heard the term of striations on the muscles, and so you'll see how, kind of how these nuclei are kind of elongated, and then with that comes the cells, the cell wall, the rest of the cell that's very elongated. And that is marked of the muscle cells. Okay, and so that's something you can always look for when you're looking for muscle cells. Okay, perfect. Let me show you then. I want you guys to look and read this. So what is the eosinophilic esophagitis? Okay, oops. So why don't you go ahead and read this? I'll give you 30 seconds. Okay. So we saw the esophagus, right? And as you probably know, the esophagitis just means an inflammation. Itis always is going to mean inflammation, and then of the esophagus here. Eosinophilic refers to the buildup of our eosinophils, which is a specific type of white blood cell. We'll talk about that a little bit in a couple moments. So now, what does this disease look like? Okay, so we had our normal here, right? We know what the kind of normal cell looks like, normal esophagus tissue looks like. Apologize for me. And now we want to see, okay, what could it possibly look like, if we have this, and how if we have this eosinophilic esophagitis. And here we can see an example of that. Now this scale is a little bit different, but the point is that you have a more closed esophagus, which is market of the condition, right? Here's this kind of this white region is where our esophagus opening would be. And then they have a blown up image here, all right? Now, what is different about this from what we were looking at? You have these bright red spots, okay? And what these are actually our eosinophils, okay? And one thing about eosinophils is that they are a type of blood cell called a granulocyte. Okay, what a granulocyte is it's a number of different cells, eosinophils being one of them, that contain these small proteins, and many of these small proteins that are released into the environment and cause inflammation. Okay, and so we can see that by taking a biopsy of the esophagus and looking at it from histology perspective, we can actually diagnose this condition quite easily, because we can see it right in front of us. Here is the problem, and here we know it compares to our normal sample. Okay. So next example I have is the lung. All right, here's an example. This is the entire lung slide. And as we kind of break this down, I want to show you the alveoli. All right. So as you might know, the alveoli are the little microscopic sacs that are inside of the lung that actually fill with air. That at the end you go through the you go through your bronchioles, then you end up in the alveoli, and your alveoli fill with air, right? We'll say air for the sake of argument, and then release carbon dioxide, right? And so this you can see here is one single alveolus. And so in the entire lung, we can see the many alveolus. And I want you to note, alveoli. And I want you to note the size of these little alveoli, okay? And we'll go to see another condition called emphysema, which you might have heard of. And so I'll give you 30 seconds to read this and think about how could this have changed due to emphysema. So read over this really quick, 30 seconds, 45 seconds. Okay, someone want to guess? Let's see. All right, so if I had this, right, this is my normal. How do you think the alveoli would have changed because of a patient with emphysema? Go ahead, unmute and guess, or pop it in the chat.
Would they get bigger?
That's precisely, yes, that's precisely it. And so here we see both the enlargement of the individual alveoli such that they are so enlarged, they begin to kind of break down, and the cells no longer form that very nice structure that we saw with that round shape of our alveoli. So that's exactly right. And that's really, one of the ways we can diagnose emphysema, as opposed to maybe another lung condition that might have similar symptoms on a kind of macroscopic level, on a clinical evaluation. But here's one way we can know for sure that emphysema is what is occurring. Well, there are other conditions that kind of cause a similar histology, but this is one really good example of how you can look at the biopsy and really make a clear differentiation between what a normal and a pathologic for a specific disease could look like. Okay, so here is an example of the skin. And I kind of want to show this for the sake of showing the different skin levels, because I think this is very interesting. If you think about, your skin, and maybe you're familiar with the kind of cartoon diagrams of the different levels of the skin, perhaps. But here we see there are very distinct levels in the skin in how the cell structure is. Okay, and so on the top here, we actually have a dead layer of cells. Okay, and that's what you see when you look at your skin and you have, dry skin that kind of comes off, for example. That's a more vivid example. If you had, dry skin, it'd probably be a little bit thicker, for example. And so certain diseases where you tend to produce a lot of dead skin cells, you'd have a much thicker region here. Okay. And so I want to kind of go down and I'll give you the right terms here. So this entire region is the epidermis. The epidermis consists of different parts. All right. So as you can see here, kind of on the right side, there are multiple layers. You have the stratum bacillus, you have the stratum spinosum, whatever. I won't go into detail about those, but know that the cells called you know what? Forget it. I'm not going to explain the cells. It's going to be too much to talk about right now. But you can read this if you would like. I'm going to post this on Schoology too, so you can read it if you want, if you're interested. But basically know that the cell type is going to differ a little bit, and what the operations of the cell is going to differ a little bit between the different layers of our epidermis and then the other layers, the dermis, and then below. Okay. So let me go to the dermis here and I'll make sure. Okay. So a little bit broader, right? But it's this region below the epidermis. And again, there are multiple different regions and you have, the sweat glands inside of it. And the next thing is the hair follicle. Okay. And I think this is something that you might find a little bit more easy to kind of conceptualize because it is much, larger, obviously relative when we're saying larger in the histology perspective. But here's an example of, your hair follicle, which is I hope everyone's familiar with kind of what a hair follicle is, but basically the region at the base of where, your hairs are. So when they sit on your skin, at the base below into the dermis, actually through the epidermis and into the dermis, you have the root of your hair, right? Which is your hair follicle. And you can see that right here. Where's my little pointer? There it is. Okay. And so here you have kind of that base of that hair follicle. And notice how it's surrounded by the cells, which we would consider the same type of cell type as the upper epidermis, which is our upper layer here, right? And so you can see this purplish color. Okay. And so that actually stretches down because you have the hair follicle here. And here is kind of the hair itself. You can kind of tell, but don't think about it too much. And then the base is where you have the generation. And so there are a lot of particular cells here that are responsible for creating that environment where the hair follicle and the hair can eventually grow from. Okay. So where am I with the next one? Red blood cells. All right, and blood cells in general. And so I'm sure you're familiar with the idea that a red blood cell is kind of like what do you call it? It's like well, you can see it, but it's got like a divot in the middle, right? And it's kind of like a boat in a sense, and that's kind of the way we represent it graphically. But here you can see this is quite literally what the blood cell looks like. And I think this is kind of cool because most students see the graphic and don't get to see, what the actual cell looks like in the body. And so don't worry, but don't worry about the purple, because what I want you to know is that inside of the red blood cells, there is no purple. And what do we know about red blood cells that would be the reason why there's no purple region? Someone give me an answer? Remember?
Do they not have nuclei?
That's right. That's exactly right. And so when you know there's a red blood cell there, right, if you're looking at the histology, because there is no nucleus, which is one of the major things that allows us to deviate from most of the other cells that we have in our body. Okay. And so that really is one of the reasons that it allows for the hemoglobin, and forget about it. I'm not going to explain it, but basically the way that it transfers oxygen, moves oxygen throughout the body and carbon dioxide and moves it around. Okay. So I want to show you some of the immune cells. First of all, platelets, which are the the cells that are involved in clotting. And what you'll notice is that they are very, very small. They are actually these purple things that we saw in the other section as well, but they're of course in the blood, and they're in much less volume, quantity than the red blood cells. But so you have an idea of kind of what that might look like on a histology. Okay. So here's a list of a bunch of cells. I want you kind of look at it briefly, and I'll explain it just so you know. And I'm kind of an immunology nerd, so I think I'm going to explain this a little bit more detailed than the other ones because I know a little bit more too. Neutrophils are our first cell. They are responsible for, basically eating cells, right? Eating our microbes that we don't want. And maybe you've heard of the concept of phagocytosis, which is this idea where you have a cell and it comes and eats the other cell. It's great. It's absolutely spectacular. And it's one thing that I think is really cool about the immune system. And so the next thing on this list, and I'll show them in a moment, is the eosinophil. And that's actually what we looked at before. And what you'll notice is that these are, like I said, the granulocytes. Okay. And so you'll notice when we look at it in a moment, is that they're filled with granules, proteins, little proteins that can eventually be released to cause inflammation, which is helpful in bringing a bunch of the necessary cells to come and kill an infection. Okay. And these are specifically circulating leukocytes or white blood cells that actually stay in the bloodstream as opposed to staying in the tissue, which there are some cells that actually, will sit in your skin tissue and they'll stay there because they are effective when you have maybe when you get a cut, right? And then you have a bunch of microbes, in your cut, and you don't want, you want an immediate response. And so by having that, the cells inside of your tissue can actually respond and immediately bring in all these leukocytes that we're looking at now. These leukocyte just means white blood cell. All these circulating leukocytes, which are in your blood. Okay, what was the next thing I'm looking at? All right, a lymphocyte. And so these are part of your adaptive immune response, which is basically you have your immediate response, and then you have a little bit later when you have a specific response to a specific antigen. Okay, so whether that be a specific microbe, a specific part of a microbe, or maybe an allergen or something. And so a lymphocyte actually adapts. It's part of the adaptive response, which evolves and then kind of responds to a infection after some time. It's a bad explanation, but I'm trying not to talk too much biology and immunology here. Okay. And the next thing is the monocyte. And a monocyte actually is a cell that eventually turns into a macrophage. Maybe you've heard that term. And that's another one of those cells that kind of eats up the microbe. And so let's go back to the top then. We'll look at that first neutrophil here. Okay, so I think notice the difference and just kind of think see how it's larger, much larger than the red blood cells. And that's one of the reasons that it's capable of what it does and one of its functions. And you notice that the nucleus is there, right? You actually have kind of like a, what do you call it? Like a bunch of kind of purple regions here that all merge together. But the basic concept is that you have multiple regions where there's DNA or RNA present. So just keep that in mind as you look at these colors here. Okay, I'm going to jump to the eosinophils because I think this one's really interesting. Notice how you have your nucleus, right? You have the region where, sorry, I was letting someone in. You have the region where you have your DNA, but then you also have this red region, and that goes back to that first slide we were looking at with the acidophilic dye that binds to the proteins. And so this is what I was saying with the granules, and these granules are actually proteins that will eventually be released in the case of an infection, and there's a whole cascade response that causes this release, but they'll be released, and then you have a bunch of immune cells come to respond at the site of an infection. But again, looking at the histology right now, you can see these granulocytes. And when we saw let me, if I can find that again. When we saw these pink things here, right? And you'll notice that the scaling is very different. It's much farther kind of zoomed. And so that's why you don't note the individual little granulocytes, but you do understand that the protein where there's a large amount of protein, where you have a large amount of that pink acidophilic dye coming and binding. And again, I know these are different slides, but the dyes that are used are relatively standard, at least for the purple and the pink that we're looking at. There's another one that we'll show at the end as well. Okay, so what's the next one on the list? A lymphocyte. I won't talk about basophils too much. A lymphocyte's a little bit smaller than a neutrophil, right? And so that's one thing you want to keep in mind, thinking about, all right, if it's a cell that's about to eat another cell, it's different than it be, different from a lymphocyte, which there are some lymphocytes that do that, but the one that we're looking at is not that type of lymphocyte, okay? So last one is a monocyte. Again, very large cell. And one of the reasons for that is because that it needs to be the one that will become what we call a macrophage. It's just another name for it when it enters a tissue, and then ends up, consuming other microbes, or not other microbes, but microbes that would cause an infection. Okay. So here is another one of those descriptions. Why don't you read this, 45 seconds. Perhaps you're familiar with the term sickle cell disease. Go ahead. Okay. So basically, what do you think? I think this is something that is explained in a very cellular manner. So you can imagine what the cell might look like, as opposed to when we were talking about the alveoli, right? Now we see, okay, the cell changes shape, right? Now, what might that actually look like? Here you can see exactly what the difference between our normal hemoglobin cells, which are round and have that kind of little dip, kind of like our boat for the hemoglobin and oxygen to be carried. And so on the other hand, you have our sickle cell, which you notice does not have the hemoglobin proper, right? And so it makes it much less effective. And you can maybe imagine how if a sickle cell is clumping together, how that could happen, as opposed to with very round cells. If you think about, if you have a bunch of balls going through a tube that are very kind of round in the shape of a blood cell or something, I guess it wouldn't be a ball then, it'd be like red blood cell shaped little things going through a tube. It'd be less likely for them to kind of get stuck together, right? Because they kind of roll off of each other and this and that. But now we see the sickle cell. It's a little bit different. It's more like that crescent moon shape that we're familiar with. It'd be much more easy for them to kind of clash and clump together is what it's called, and cause issues in the vessels if you have lots and lots that clump together over time. And so, looking at this example of the microscopic level, you can kind of have a better perception of that than maybe without having learned this histology. Okay, so the last one I kind of want to show is just the brain. Okay, I say just the brain, like just the brain. It's not that important, but it's very important. Don't worry about the names of these cells. They're just different types of neurons. They're just different names. But here you have something called Golgi staining. All right, you'll see it at the top, actually. This is a rat's brain, and it says using Golgi's method. And what the difference is that we're staining a very very specifically staining the neurons. All right, and what you might know about neurons, and if you aren't familiar with the structure of neurons, which I don't expect you to be, you can see on the right here, basically you have the cell body, okay, the main part where the nucleus would be, and you don't have that purple staining here because of this. They specifically wanted to show the neurons, but you can see when you have that on the right side there, you have the cell body, which would have the nucleus. But then from that, you have both on one end, you have the dendrites, right, which are very clear in this graphic, in this image here, in the histology here. and which kind of branch out and transfer the message, which is received from the other end of that cell body, which is called our axon. Okay, and that's a little bit wider generally. It's hard to kind of conceptualize, but you can see the dendrites very clearly. Maybe you can kind of make out the larger splotches too, which we can consider our cell body. Okay, and I just think this is kind of cool to look at, and then I'll show you the pyramidal cells as well. And this is even more clear to see, okay, right there. Where's my little, I like my, there we go. All right. I got, you got the cell body here, this very, kind of thick region. And then you have a thicker axon this way with your dendrites all branching out and very thin in the other direction. And obviously, there's a huge amount of overlap. And that's why you have the different kind of, levels, where there's a bunch of different cells. But as you might know, the brain, that is what the brain essentially is. It's all these neurons that all fire and I will say work together to achieve a message and achieve action. And then we, eventually will transfer down the spinal cord, which we saw as well. And so that was just so you get a basic overview of what this idea of histology is. Maybe you've heard the term, maybe you've heard of microanatomy, which is the exact same thing in this example. But I hope this gave you kind of some insight into kind of some of the cool things that you'll look at in the future if you go into medicine and learn about. So does anyone have any questions or anything that they want to maybe look at more, a specific organ? I don't know if I can answer all questions because I don't know everything, but go ahead and ask. Oops. All right, so if you have more questions, then I think we'll kind of wrap it up. I hope you found that interesting. I know we had a smaller group, but I appreciate you guys coming. I know it was a day off too, so I hope you enjoyed what we talked about and learned something new, because I feel like you're getting an experience that most high school students would never even touch, this idea of histology for a very long time, for 4 or 5 years from now. So yeah, thank you for coming. Free to go. Stick around if you have questions, anything to ask, and we'll see you next week.